I will. I know this is annoying, but I will be starting and stopping recordings also during this morning, so that we can. So you don't have to listen to like a 2-hour long recording to find find one little bit of information. Okay. All right. And I hope, and I have to tell you too, I hope Todd and Alyssa will remind me if there are things I forget. I made sure to write everything down, so hopefully I'm not going to forget everything. Sorry about reading all these things to you, but I just know if I didn't write it down, I would definitely forget things. And this link, by the way, is also available in Slack. I'll come back to Slack in a minute, but I first want to thank our sponsor, our funder, the National Science Foundation's Advanced Technological Education Program, also known as the ATE program. And it's their grant that's paying for this and will be funding us through the next 3 years, so we can have a couple more hackathons. As you've been figuring out, we're going to do a lot of work through Slack. So I'm going to stop sharing my screen for just a minute and go to Slack to show you where you can find things on Slack. And I will remember to share my screen and share the Slack screen. Please let me know if I forget to do this, because I do forget to do some of these things. I'll warn you right now. Okay, so in Slack, there is a bar at the top where you can search for things, and you can also look for messages and files and channels and people. Okay, there are threads where people have had conversations. You can select direct messages where you're conversing with a certain person. If you want to write drafts, you can do that. If you want to save items, you can do that and find it here. There is a link here that says more, and you can use that to find things you don't see, like a channel browser will help you see channels if you don't see them, or people, and files. Okay, I want to just point out some of the key channels here. Each team has their own channel. If you haven't joined your team's channel, then be sure to do so at some point, and your teams will be conversing with each other in their channels. If you want to listen in on another team's channel, by all means, we are all here to learn, and you can learn not just from your own team, but you can learn from the other teams as well. The really critical channels outside of your team channels here are announcements. And this is where we will post information about what's going on. As you see, Alyssa's posted that we started our kickoff. In the announcements, you'll see some of these are kind of yellowish. Yellowish announcements are ones that are also pinned. And pinning is a thing you can do if you want to make announcements easier to find. It kind of highlights them yellow, and then they show up at the top here. So I have 4 announcements that are pinned, and if I just click pinned, I will see them here. Okay. I can also see them just by scrolling. So things that are pinned here, we're using the same Zoom link for each meeting day this week. You can find it here in the passcode. And I have the schedule. I have put some links to things in the schedule. That link that says kickoff and introduction are the notes I'm showing you. Another link here is going to be from when I talk a little bit later about antibodies and IC and 3D. And I'll probably put some more links in here as well as the day goes on. Okay, so announcements, you're going to find a lot of materials, resources. I also put in here, I thought we might like to have a glossary. People in this group are going to have differing amounts of experience with antibodies, and I thought it might help to define some of the terms. I don't have very many definitions in here, but if you have a definition you want, you can add it to this, or if you have a definition you think people should know, feel free to add it. I put in some of the basic ones here, antibody, antigen, epitope. So you've got those. I also put some structures that you might want to use to get started. A mouse IgG, a human secretory IgA, and a human IgM, and one that I annotated with rainbow. And you can see if you just click this, actually, It doesn't share it, does it? It doesn't share it, but after a while, yeah, well, I can do that. I'll show you what that looks like. Okay. There we go. Isn't that pretty? So there's a human IgM that's been annotated. I think one of the most powerful features of IC in 3D, and it took me a while to appreciate this, I have to admit, is that you can save lifelong links to structures that you've annotated, or your students can save them. And I think that is a really helpful thing. When I was grading this fall, I was really glad to have the links from my students to be able to go back to the structures that they were working with and go, okay, so that's what they did. They did look at this. They did see this. They did annotate it. All right. Anyway, any of you can click those links and you see those structures. Okay, back to Slack. All right, so that's one of the channels. The other channel in Slack, I just want to point out, is the help desk. And if you're having any troubles with things, you have questions with things, you're really lost on Slack, or you need to get a hold of somebody, go to the help desk and team leaders will be monitoring this. Or if you have a question about how something works, we'll be monitoring this throughout the hackathon. Okay, any questions about Slack right now?
Sandy, could you actually go through how they can find and add channels? That would be probably helpful.
Okay, all right, good idea. Okay, so this link here that says more, if you click more, there is a channel browser. And if you click channel browser, you will see all the channels that are here. And now I have joined all the channels, but let's see, I'll leave a channel and then I'll come back to it. So I'm leaving one. And if I do not belong to a channel, when I click it, I see this option to join. Okay. So now I've joined and it will appear in the side. And if anybody is posting messages in that channel, it'll be highlighted. Does that help? Other questions?
You can also click on the hashed links that I posted in Slack that's pinned. So if you really can't figure out how to add the channel for your team, you can go to the pins in the announcements channel, and you can also just click on your team name and that'll also ask you if you want to join.
Yeah, and the pins that Alyssa is talking about, if I open the announcements channel and I go up to pins, I will see that Alyssa, here are all the team channels. So you could click any one of these and you would get to that channel. Thanks. All right.
Yeah, I can comment. Since this is our first hackathon, if you have any difficulty, don't worry about using the help desk, because we want you to do that, because that's going to be giving us feedback throughout the process that will make the next ones just that much better.
And oh, go ahead.
And even if you don't have a question, please join the help desk, because you may have a question, somebody else may have a question that's more like bio-based or, antibody-based, and you may be able to answer it for someone else. So the help desk is not just meant for like us to help you, but also for you all to help each other.
Yep.
And I have to, oh, go ahead.
Okay. I was just going to say, as to joining other channels, yeah, anything that interests you, please do join, because I've learned in hackathons that there's things that I didn't know I knew, and you'll see a conversation and sometimes you can contribute to it. And so that's part of what makes it cool. So.
Or sometimes things you thought you knew, but you really didn't. Sometimes Slack is one of those for me. Like I said, this is a learning experience for all of us. Okay, I'm going to go back to my notes here. If I can find them. Okay, so now we've talked about Slack. I want to mention that we are using a free Slack channel, which means that it's not going to last forever. I mean, I think we can probably use it over the next 3 years, but I don't think Slack has a limit on the amount of information that it saves for people that are using free channels. So when it gets to a certain point, I think it deletes the older stuff. Is that right? Yeah.
Right. Yeah. So it'll exist for as long as Slack is free, but you may not be able to find old messages. So the workspace itself, for as long as Slack has a free mode, it will be there and you can chat. But unfortunately, like once we've hit 10,000 messages, if you wanted to go back to something really cool from like 5 years ago, depending on how much we chat, it may not be there anymore.
Exactly. And files, I think, are the same way. So we are also going to be using another system for storing the information that we work on and produce through the hackathon called Cubes. And I am really new to Cubes. So I know I've asked a whole bunch of you to join, and you're probably still waiting for your invitations, to be accepted and to be added to a team. Still kind of figuring that out. We will have one of the people from Cubes joining the writers meeting tomorrow to help out and answer questions about it. Cubes is kind of has been funded by the NSF for a long time. And it's sort of, for those of you who've been a little familiar with software engineering, it's sort of like a GitHub kind of thing for education. The idea being that groups of faculty people can work together in Cubes and they can publish what they produce, that those publications can be adapted by another group who can add to them and continue on with them. And I'll talk about that a little bit more later on. Okay, I also set up a LinkedIn group called Antibody Engineers. The idea, and this links to it also, feel free to join it. The idea being that we can build a larger community and eventually people might share job posts and other interesting things through here, articles and things like that. Okay, so the schedule today. We have a schedule in the website and on Slack, and I'm just going to go to that real quick and walk through it. So I'm on the website right now. This is the Hackathon January 22 page. We do have links here to our code of conduct, by the way. If you haven't read that, you should, but I'm not going to go through that right now. The helpful information and background reading, which we may be expanding, and example team roles, and the schedule. All right, so I put all the times in Eastern time. I know time zones are confusing, but it's good to stick with one. Today, we're going through the logistics. I'm gonna talk about antibodies and introduce IC and 3D. Dr. Erin Dolan, who is an expert on course-based undergraduate research experiences, is going to talk about cures. Marcus Mendes from the La Jolla Institute for Immunology is going to talk about Iedb. David Bloom on our advisory board, I should mention too, is going to talk about antibody manufacturing, as is Dr. Frances Weiss Garcia from Memorial Sloan Kettering Center. She's talking about antibody validation, which is really, really a critical thing for making sure your antibodies are doing what they're supposed to be doing and not doing what they're not supposed to be doing. After that, your teams are going to, I'm going to open up the breakout rooms. I've got a breakout room for each team. So you'll meet with your team and figure out if you're going to meet right away or meet later on during the day, but you'll plan a time to meet and you're going to work on planning your project. And I'll come back to that in just a minute. And the rest of the day, you have time to work. Tomorrow morning, each team is going to present their plan for what they're going to work on this week in the hackathon. I have 15 minutes each, but it doesn't have to be that long. It can be shorter. The writers are going to meet at, again, this is all Eastern time, so 12:30 Eastern time. There'll be open work time, and the teams will meet whenever you'll work out when you're going to meet. Saturday, we'll have a quick check-in to see if there are any problems, and each team will let us know what's going on, and if there's questions they have or things they need. The writers will meet, you'll be working, and then Sunday, every team is going to present their results, and I have an evaluation form for you to fill out. So that's the general schedule. What are your teams going to be doing today? Well, today, you're going to meet the other people in your team and introduce yourselves. You're going to be brainstorming your project plan. You're going to be choosing your team roles, like who wants to be the writer, who wants to be a subject matter expert, who wants to be the technical expert. We have the team leaders already set. Some of those roles, by the way, can have more than one person working on them. It's just it's nice to have roles so that people can coordinate their work. So one person is in charge of writing, other people can, talk with them and get that worked out. Your team will set their meeting times, and your team will figure out what you're going to do to present your plan tomorrow. Questions so far? Okay. So what are we trying to do here in this hackathon? Well, we really have kind of three main goals, and I have to thank Alyssa for describing this to me because it was really helpful. Oh yes, there is a link to the documents I'm showing. This one is in the schedule in the Slack channel. It's the one that says kickoff is linked. Actually, you know what? I will just quickly go to Slack and show you. And maybe I should even check it, right? Make sure. I'm pretty sure I put it there. So announcements, all these things are going to be in announcements. Schedule. This one is the kickoff and introductions. It's right here. And it's also pinned to the top, but I find some of these easier to scroll. Just look for the yellowish kinds of things. Okay, back to the document. All right. Okay, so right, so what are our goals? Well, we're going to be creating things, right? Building things, which is fun. We're going to be networking with each other, and I hope everybody has fun during the process. And the things we're creating are research projects that can be used by classes to have students do undergraduate research related to antibody engineering. What kinds of things are you going to be putting together to make those research projects? Well, you're going to want to have what questions you're going to address, what kinds of background information and references you might need. For those of you working on lab projects, you're going to want to have some kind of lists of materials and where to buy them, what they're going to cost. We do have some money for lab supplies, not an infinite amount, but we can help with some of this as after the hackathon, as the work is going on. There'll be lab protocols. Like if you're making an antibody and you need to validate it, how do you stain it? How do you know it's working? The process you'll follow, since these projects would be used in a class or maybe even outside of a class, many of your faculty, you're going to need to know how to assess the project. So come up with some of those. And then it would be helpful to have a list of skills that will be learned, not just the technical skills, but also the soft skills, because you'll want your students to be able to put those on their resumes at some point after they do the projects. So how did this project come to be? Well, for the past few years, a lot of publications have been coming out demonstrating the benefits of undergraduate research. And it hasn't always been a thing that students at two-year colleges have been able to do. So I think it's really great that it's becoming more prevalent. But a lot of, to do a long-term, year-long or so research project, sometimes that works in community colleges, sometimes it doesn't. So And even the larger colleges sometimes, or universities, sometimes you can do it, sometimes you can't. So there's been this movement to have alternatives that are shorter projects that could be kind of plugged into a course. They could be longer, they could go longer, but they get started maybe 1 or 2 weeks in a course. So this is where the word, the phrase course-based undergraduate research experience has come from. And there are, there's actually databases of some of these. And I've looked at them, and they're really interesting. There's, all kinds of things like, I don't know, barcoding the world, or tiny earth, looking for antibiotic resistance and some of these things. But what I saw is that I didn't really see things that were related so much to biotechnology. And I didn't see many course-based undergraduate research projects that were applied research. That is the kinds of things that you might want to do in, thanks, Christine. She put a link to some of the projects here in the chat. I didn't see things that were applied. So skills that would be useful for students who want to go into biotechnology. So all of a sudden, I was at this talk a couple of years ago by this guy named Just Biotherapeutics, was talking about antibodies and some of the issues with antibodies. And I just realized antibodies would be a really cool thing to have as a focus of undergraduate research. Now, why? Well, one, I think in research labs, they're ubiquitous. All kinds of people are using antibodies as research tools. They're using them for staining, for flow cytometry, Western blots to follow proteins and understand what they're doing, to precipitate proteins and block activity. In biotech, antibodies are really prevalent as diagnostic tools. You might know if you've ever had a home pregnancy test, or probably not all of you have done that, but some people have. And if you're taking any home rapid SARS tests, even the tests that have antigens are relying on antibodies. So they're really important from that standpoint. And therapeutics, I was just astounded to see that if we look at the top selling drugs, I just did a search for that from Genetic Engineering News. And if I go straight there, I can see that a whole bunch of these are antibodies. Now, how do I know they're antibodies? Because there is this 3-letter suffix that says MAB, MAB, short for monoclonal antibody. And these were the top 3 drugs in February 2020. That's a monoclonal antibody for treating thyroid eye disease. Here, not that one. Here's one, Ruxience, a monoclonal antibody for treating non-Hodgkin's lymphoma. Here's one for treating breast cancer. Here's one for treating vaso-occlusive crises, sickle cell disease. Okay, sickle cell. And here's another one. Oh, this one we're going to talk about a little bit. This is the cocktail from Regeneron for treating people who are sick with COVID. Not working anymore, but it was very important. And some other famous drugs are also antibody-based. You may have heard of Herceptin or Avastin or Humira or Opdivo or Rituxan. Our granddaughter took Avastin. She was born prematurely, and she took Avastin to prevent blood vessels from growing into her eyes. So there's kind of some amazing uses. They're really powerful drugs. But the problem is that is that antibodies are proteins, and proteins are not as easy to make into drugs as small molecules. Drug companies can take a small molecule and they can package it in a pill, and sell it at the drugstore that you can get and take it home. But antibodies don't work like that. Antibodies are bigger. They're complicated to manufacture. Because they're complicated to manufacture, they're expensive. And only some countries in the world have access to antibody drugs because of the expense. So if our work can figure out how to make them cheaper and easier to manufacture, that is a good thing. It's also challenging because with antibodies, like I said, you can't make a pill out of an antibody. It might get destroyed in your stomach if you take it. So people who are getting antibody treatments, a lot of those treatments are chemotherapy. Those treatments are administered through IVs. And a lot of hospitals and clinics, although they're set up to administer chemotherapy to cancer patients, they're not necessarily set up to administer those treatments to other people. In fact, it's been a problem for people who want to get monoclonal antibody treatment when they have COVID, because not all hospitals are able to do this. Okay, so antibodies are cool, right? We can do a lot of things with them. So why a hackathon? The reason I thought it would be good to do a hackathon to come up with our research projects related to antibodies is I really want to involve a lot of people. All right. I think the more people we have working on these ideas and working together, the more likely we are to come up with some really good ideas and some ideas that work for a lot of people. Plus, I found, you might not be aware of this, but ICN3D itself has been developed through a series of hackathons. And we're going to hear how useful a tool that is in a little bit. Uwe, I will look for some, there's a question about steps in therapeutic antibody development. I will find some of those and make those available later on during the course of the hackathon. It's a good question.
Thanks.
Okay, so hackathons. I attended my first hackathon last summer, and I found it to be an incredible tool for learning and a really kind of immersive learning experience that I hadn't really expected. I had no idea what was going to happen going in. Can't believe I wrote a grant on doing it, but it was great. It was great. And I really enjoyed it. And I realized some things from it that I hadn't appreciated before, which is that I think hackathons are a really good way to model collaborative science. We have this myth, right, that science is a whole bunch of people working all by themselves in a lab. Maybe the lab works a little bit together, but I know when I was a graduate student and when I was a postdoc, everybody in our lab each had their own little project. And boy, you better not encroach on anybody else's project. Okay, or you were in trouble. And even though we would share information and share ideas somewhat in lab meetings, it was always really competitive and cutthroat, and it wasn't fun all the time. And I think it could have been so much better had we actually tried to work with each other. I mean, one of the things I disliked most about graduate school is that maybe there were 10 people in the world that cared what you were doing, and you couldn't talk to any of them. And there's been a big movement, I believe, I understand, to make science more collaborative. And I think by working together in hackathons, we can model a new way of doing science and working together. Not only that, if those of us who are preparing students to go into the workforce really need this kind of practice, because in the workforce, people work together. Companies can't make products and get them out to the world, no matter what kind of product they are, whether it's cultured meat or antibody drugs or algae dyes or biofuels. They can't make products unless people in the companies work together. And this is actually a really important soft skill that we're always hearing from our employers that they want. So I think hackathons and the kind of thing we're going to be getting practice with is going to help us get better at doing this. At least that's a hypothesis. Okay, yeah, so this gives us practice for opportunities to practice soft skills. I know there are some students participating with us, and I'm hoping that we'll see more students participating over the course of the next 3 years. They get to practice leadership, accountability, communication, and I'm hoping all student members who participate think this is a good thing to put on your resume. Okay, you participated in the hackathon and this is what you did. I also think hackathons have some potential for overcoming some natural problems that we have with curriculum development, which is, at least in the virtual sense, which is that if you have a workshop and you have a few faculty come to work on something together, you can't really scale it. I mean, if you're traveling, there's only so much money for traveling. If you are working together, it's hard to do things and grow. I expect that this will expand because we have a certain number of team leaders now, right? But next year we can have more. If you have a project that you come up with that you want to be a team leader on, that's the limiting factor, right? Is the number of team leaders. So I think this is something that can grow. Again, it's a hypothesis. We'll see. We'll see how that works in reality. Okay. And I also at some point would like to include mechanisms for students to present projects. And Todd, you look like you want to say something.
You don't I? I was gonna comment. One of our advisory board members from Molecular Mold Tech, she agreed to join us because she hired people through hackathons. So so she saw that hackathon concept and happened to be working on antibody engineering. And when we asked, she's very enthusiastic about this. So it's a really useful format for getting people engaged and all the things Sandy just said. And sometimes people get hired.
Right.
So to be fair, in industry, hackathons run a little bit different than how we run them, but like basically their sole purpose is to figure out which of them have the tech skills and the group skills to be hired for their companies. So those headhunters often at more like industrial focused hackathons.
Okay, well, I have to say this is still new to me and still new to almost all of our team leaders. And I know from reading your applications, this is new to many of you as well. So we are going to work hard to make this a good experience for everybody. Okay, yeah, I mentioned before that I see this kind of these kinds of events as ways to model new avenues for curriculum development. And I mentioned cubes, and we will be learning more about cubes, I guess, probably tomorrow. Okay, so a little bit more about logistics. All of you are going to be working in a team. You already know we're communicating through Slack and Zoom. I didn't mention this, but I think I might have put it in the schedule that the Zoom rooms are open like all day. And we're using the same Zoom link every day. So you should be able to access, not the Zoom rooms, but the Zoom is open all day and you should be able to access it. I think I have to like name the rooms each time. So if you come in and join and there isn't a room for you, you can probably just take a room and be in there. Right now, though, there are rooms and I will open them after we have our talks this morning. Okay, I mentioned things will be recorded. You can join other channels. Yeah, team leaders, why don't you say a couple words about your projects? I know you already introduced yourself, but just mention which project you're on and yeah, what you're leading. And why don't you start, let's see, Sheila, why don't you start? Oh, you need to unmute. Yeah. Okay.
Oh, okay. All right. So we are going to start with the Iedb project. And like I mentioned to you that I had a great opportunity to watch Sandy do a webinar. And from that webinar, we were able to get some student ideas on how to run a student project on a topic. And the part that I have really used for the Iedb that our students really liked was the Discotope. which is looking at the B cell antibodies. And our students were very surprised to see some of the assays that we had introduced in the Iedb powerful database. But there was one area that I really realized coming from the freshmen and the sophomore students who are part of our introductory biotech classes in microbiology, is that they needed a glossary. And the glossary was super extensive. It wasn't even what the glossary that you were talking about. We had to start from amino acid structures, primary structure, secondary structure, tertiary, and go all the way up to antigen, epitope, paratope, all of those. So I'm in the process of creating some short 2-minute videos so that I can scaffold some of these research projects so that students can really have an understanding of what's going on. So we wish to present one of this at our local DOE presentation, poster presentation for community college students in Illinois. So that's sort of the goal at where I am.
Cool. Nagy.
My project, or my team's project, is break an antibody. So why would you want to break an antibody? So this came from sort of a common practice in biotherapeutic development, and that is to switch out one or more amino acids to enhance the antibody product, enhance the properties of the antibody product. And that might be to enhance binding to the target, to make it more stable, more resilient during production, or even to stop it from aggregating after downstream processing. And that was the case with the antibody that we produce in our lab. It's an anti-interleukin-8 monoclonal antibody. And that had been modified genetically and then, protein-wise to be resistant to aggregation, which can be a problem and something that the FDA requires you to test for. So thinking along those lines, we thought it'd be important for students to understand how you design those changes and then how you actually make them happen. And then subsequently, how do you test for that you've actually successfully made the change and enhanced the properties. But talking to our advisory board, they mentioned that it can be complicated to evaluate these changes if you're just enhancing a property of an antibody. Why don't you just knock out the function? That's something that we can evaluate much simpler, more simple or easier. So that's what we decided to do. So we'll be designing a project where we will have to, choose an antibody to work with, modify the CDR region, and then once we've made the modifications using bioinformatics, we will then work out the actual process to do that in the lab. So we'll be researching, components that we have to purchase in order for a student to modify an antibody genetically, and then purify the antibody and test and evaluate that we've actually made the change that we think we've made. So that's the idea behind our Break an Antibody project.
I think that's so cool. I actually found a paper on breaking one the other day too. It's a little nanobody that binds to zinc, and they were breaking it in order to understand which residues were most important in zinc binding. I'll see if I can find that.
Yeah, nanobodies might be the way to go. A little simpler, maybe.
Yeah.
Mm-hmm.
Okay, Aaron.
Hi, the project that I'm leading is the affordable antibody engineering, and this is motivated by the background. Essentially, many biotech training program, even though we're training students to work in the pharmaceutical, and as Sandy pointed out, one of the major product of pharmaceutical industry, biopharmaceutical industry, is antibody, monoclonal antibody. And unfortunately, many biotech training program did not they have no ability to talk about or train student in terms of how the antibody is designed and produced, right? I mean, for several reasons, right? Because the concept it's it require very high barrier of entry, right? In order, the most common practice in the industry is you do like high throughput screening method, and that require really expensive equipment like cell sorter, things like that, and many biotech training program, we simply don't have that funding to afford that kind of equipment, and this is why we're trying to develop some sort of like experimental workflow that can still allow educator to sort of like give student overview of what's happening during in this high throughput fancy antibody development, so that at least coming out from the program, they know what the process is. So that's of course we're trying to develop. I mean, the keyword for our project is affordable. So everything has to be done in the budget scale that perhaps reasonable for typical community college biotech program. So
I appreciate that part, Aaron. Yeah, I think that's going to be pretty neat, because imagine people could be making their own antibody reagents for their classes. It'd be kind of
and
cool.
I think this one will sort of like synergize with what Maggie's team is doing, right? So the principle of breaking antibody, the computational part of that one is somewhat similar, and then we can always combine it with the screening, affordable screening method that we developed. So,
all right, Todd.
Yeah, we're going to work with ICN 3D. So Sandy's going to show us a little bit more about ICN 3D, but what kind of motivates this project is one, students entering the workforce in biotech these days are going to be ever increasingly working with software, instruments, software, and data. And, so you have experimental processes that are going to make that data. And so they need to understand controls and these kinds of things, but they need to be really more facile with software systems and data. And underlying that, if you look at like COVID news, right? We keep talking about these variants, Omicron, these variants. What do variants mean? What does it mean when you change a DNA sequence with respect to the structure and function of the protein? So a tool like ICN 3D brings those three elements together so that we can connect sequence to structure to function. And so that's the framework. ICN 3D is a new program, and Sandy showed, kind of a list of things for the laboratory and how we need to make protocols, lab protocols, source reagents and materials. Well, you could just substitute for software reagents and materials for data sets. So you want to teach a concept. What data do you use to teach that concept? Laboratory protocols, well, we call those user manuals in software. So what we want to come away with is some projects that could combine different data sets to teach a principle, and then the best practices for using ICN 3D in the classroom. So sounds simple, right?
Oh yeah.
and in our Slack channel, I posted some things that we'll want to focus on and pay attention to. We're going to learn how to submit feature requests and bug reports as we do this, which are really important for working with software teams and getting things working in the right way for your particular purpose.
And I have to say, we are exceptionally fortunate that we have the ICN 3D developer, Jiao Wang, who is here to help us and answer questions. And if there's anything, thank you. Jiao, can you just introduce yourself quickly?
We knew you'd be eating.
I'm sorry.
Hi, my name is Jiao Wang. I'm a staff scientist at NCBI.
And he has children.
So I'm working on the web-based 3D viewer ICN 3D, and it's released in 2016, but we are still actively developing the tool. So hopefully with this hackathon we can make it easier to use in the educational section. Yeah.
Thanks. We really, really appreciate your participation. This is great. And oh, right, I'm a team leader too. My team is focused on looking at commercial antibodies, although it doesn't have to be just commercial antibodies, and whether or not they'll be able to handle different SARS-CoV-2 variants. This project started out this summer when I was working on the hackathon this summer, because I teach bioinformatics every year for Shoreline Community College. I have a 5-week class and I do new things every year. And I thought, oh yeah, there might be variants, and maybe we could look at the antibodies and maybe we could see if the antibodies can handle them. I had no idea how timely that project would be. And so I will be able to share with you the project that I had my students work on last fall, and but we'll be able to go farther and do some more interesting things from there.
Sadie, can I add?
Yeah.
For the teams, we're going to get together. We put in project names that are descriptive of what we hope to accomplish, but maybe you come up with more of a mission and a better name. Feel free to rename your project. So
but not the channel.
When we did the hackathon this summer, ours was the effect of mutations. Can we use IC3D to understand how a mutation in a protein sequence, a DNA sequence leading to a protein, can change that structure and function? Well, that's nice, but what we really realized with SARS-CoV-2 is we were working on a molecular century system. And so we renamed our project Molecular Century to capture that idea that if we have an early warning system for variants in a virus that are affecting antibody binding, we could actually, ahead of when it's really, at pandemic level, maybe identify things as early warning. So.
One of my favorite hackathon team names, and I'm biased because it was the team I ran a long time ago as a grad student, was about an RNA-seq pipeline, and we called it Hassel. And our mascot was actually Hasselhoff.
What is that, Alyssa? What's a Hasselhoff?
It's the actor from a long time ago from Baywatch.
Oh, Baywatch. Yeah. I thought maybe it was a Pokemon thing.
No, but that would also be fun.
All right. Does anybody have any questions before we continue on? I do have the chat transcript. I will post links in Slack. I mean, not like instantly, but we'll get them later on this afternoon. Christine, did you have a question?
I do, for Todd and Jayo. I'm sorry about the noise in the background. I'm in the library. They might have students that use a program called Yasara, and they're comparing variants of unknown significance. And so how similar, if you're familiar with that, is the ICN3D? Because we're having some problems with one of the students having an older computer, and then she's having issues because it's taking too long to go through the imaging.
Well, I'm just looking at the website now. So that's it's new to me, but the concepts of displaying variants and mutations on proteins is a capability in ICN3D. In fact, there are algorithms in there from Professor Barry Honing on if you put in a change in amino acid, how is it going to change the interactions of that amino acid with other amino acids in either a protein that your protein is binding to, or intramolecular kinds of interactions. So, so the short answer is yes, I think we can probably do stuff. Jaya's nodding, so I think I gave a good answer.
Yeah, and I think we'll find out.
Yes.
Savita.
Hi, I was just curious as to why this is named Hackathon. Like I didn't know what it meant, or, I mean, I just was curious.
Because it's the name I've heard.
Okay.
So
yeah.
a hackathon is just referring to the idea that you're trying to hack at a problem, right? Like the expression life hack, it's approaching the problem with a different angle and lots of different people with different backgrounds.
Okay, thank you.
There is actually the word marathon part of that, which then alludes to one of the bullets that was on the initial introduction list on Innovate Bio, and that is, do we have to be 24 hours there? And usually in hackathons, the answer is yes. So I'm going to see how we're going to handle this here together.
That's right.
So the hackathons that I have ran have never been 24-hour events. They're usually 3 days or 4 days. When it's in person, 3 or 4 days, 9 to 5, like a normal day job kind of situation. Because what we have found is if you run 24-hour hackathons, usually the only people you get are single male students. If you make the time a little more flexible, you get more diversity in the people that participate.
And hacking, you know, it's developed a negative connotation, but really in software, it was about innovating and taking and creating new ideas and creating new things. And so really it's a focused session on creating new things is really the blending of those two words together. And so now people are starting to, kind of, because of how words can start to pick up negative tones, changing it to codathons, this and that, but we're sticking with hackathon.
But we also wanted something different than a workshop, because I've been attended and presented and organized a lot of workshops where it's just people sitting there listening to things and not actually doing anything.
Or they're hands-on, but it's more of a kind of education setting that, people are familiar with in terms of present and learn, whereas we're going to present, learn, and create and learn some more.
So whether it's the perfect term or not, it's what we've got.
It's really about that creative iteration, right? That networking and creative iteration, which is really kind of unique, and that you don't necessarily get with other types of events, at least not that spirit anyway.
Okay. Other questions right now? All right. There will be more opportunities. Right now, what I'm going to do is stop